Hi everyone, let's continue with the last lecture on bacterial growth and nutrition. The first half of the lecture will focus on biochemical factors of bacterial growth. Okay, let's look at the essential nutrients for microbial growth. So the nutrients for the microbial growth can be divided into macronutrients and micronutrients. So macronutrients means the nutrients that needed in a larger amounts, which is including carbon, nitrogen, hydrogen, oxygen, phosphorus and sulfur. So usually hydrogen and oxygen are common where they obtained from the gas, and on the other hand carbon, nitrogen, phosphorus and sulfur we must provide in the medium. Within the macronutrients, there are also some nutrients that are needed in a smaller amount, which is including calcium, ferrum, magnesium, and potassium. So these are we call as a mineral salts. And on the other hand, micronutrients, they're only needed in a trace quantity, or we call as a trace elements, which they needed in a very tiny amounts, which is including zinc and magnesium. Okay, first is the carbon. So most bacteria use carbon-containing compound for two major reasons. So first, to assemble carbon into new organic compounds, which is including amino acids, fatty acids, and organic acid. And also some bacteria use carbon as an energy source, where these bacteria we call as heterotrophs. So regarding heterotrophs, we will learn more in the following lectures. Okay, the following one is the nitrogen. So all organisms, including microbes, that need nitrogen to synthesize enzymes, proteins, amino acids and nucleic acids, and nitrogen also found in the peptidoglycan. So there's also some bacteria we call as a nitrogen fixing bacteria or cyanobacteria that can use gaseous nitrogen directly from the environment. Okay, the following one is the sulfur. So sulfur is required by organisms because of its structural role in the amino acids such as cysteine and methionine, and in a number of vitamins, which is including thiamine and biotin, and in coenzymes. So coenzymes are organic molecules that are required by certain enzymes to carry out the catalysis process. And microorganisms usually obtain sulfur from the inorganic salts. Okay, phosphorus. So phosphorus in nature occurs in organic and inorganic forms. So microorganisms can obtain phosphorus mainly from inorganic phosphate ions and use them to synthesize ATP, phospholipids and nucleic acids. And bacteria also use organic phosphates using the enzyme phosphatase. So phosphatase is an enzyme that uses water to remove a phosphate group from the protein. Okay, let's move to micronutrients, or which is also known as trace elements. So many microorganisms require a variety of trace elements such as copper, iron, zinc, cobalt, sodium, potassium, calcium, magnesium, and manganese. And usually these trace elements required in the iron form. And these trace elements also or usually act as a cofactors in the enzymatic reaction or used to activate a certain enzymes in the microorganisms. Okay, let's look at the trace element of magnesium. Okay, magnesium usually required to stabilize ribosomes, cell membranes and nucleic acid in the bacteria. And magnesium also required to activate enzymes, especially those involving phosphate transfer. And the second one is the calcium. Calcium usually did not require or not essential for the growth, but then it requires to help stabilizing the cell wall and also to provide heat stability to the endospores. Okay, following one is the sodium. So sodium is not required by all bacteria, usually is required by marine microorganisms. And iron is usually required for respiration, and it is a key component of cytochromes and iron sulfur proteins in electron transport. And cobalt is needed only for formation of vitamin B12. And while zinc play an important role in the many enzymes, which is including carbonate dehydrogenase, alcohol dehydrogenase, RNA and DNA polymerase, and other DNA binding proteins. Okay, copper. Copper play a role in certain enzymes involved in respiration like iron, and it also found in certain superoxide dismutase enzymes. And then manganese is an activator of many enzymes and is also found in certain superoxide dismutase enzyme. So superoxide dismutase is an enzyme that helps break down potentially harmful oxygen molecules in the bacteria. And followed by nickel is present is most dehydrogenase enzyme. So dehydrogenase is an enzyme that catalyzes the removal of hydrogen atoms from a molecule in the electron transport chain reaction. Okay, let's look at growth factors. So some bacteria, they are remarkable, which means they're able to make all the organic compounds that are needed from a single carbon source like glucose. But then some, they can't. So therefore, we need to add vitamins, amino acids, blood, or other things into the culture media for them to grow. And the vitamins, amino acid, blood, and etc. we call as a growth factors. And bacteria that require a medium with various growth factor and other components are hard to grow. And then these bacteria we call as a fastidious bacteria. And these substances usually require a small quantity and usually they cannot be synthesized by the bacteria. The substances frequently serving as growth factors are vitamins, amino acids, purines, and pyrimidines. So the next subtopic is the nutrient uptake in bacteria, or in other words, how bacteria eat or microbes eat. Okay, the nutrient uptake in bacteria can be passive transport or active transport. The major difference between passive transport and active transport is requirement of ATP in the active transport. And on the other hand, passive transport can be divided into passive diffusion and facilitated diffusion. Okay, passive diffusion happens when a molecule moves from a high concentration to low concentration gradient, basically using a osmotic pressure. And then many of the material movement in the cell done by this way, which is including osmosis and dialysis. Okay, the next passive transport is facilitated diffusion, and it is a process facilitated by integral membrane protein. And the integral membrane protein is a transport protein which integrated into the cell membrane, and usually these proteins are highly selective in allowing material to cross. And for example, glucose is enter most of the cells by facilitated diffusion. Hence, as for now, remember that osmosis, dialysis, and facilitated diffusion are examples of passive transport because they are not using ATP. Okay, the last one is the active transport. So active transport is used to transfer bigger molecules and ions across the cell membrane. Because these molecules are too big, therefore they cannot penetrate through or diffuse through the cell membrane. And usually in order to diffuse them or penetrate the cell membrane, they need energy or ATP. And then just remember that active transport is responsible for cells containing relatively high concentration of potassium ions but low concentration of sodium ions. Okay, this is a summary of nutrient uptake. So if it's a passive transport, which is including passive diffusion and facilitated diffusion. So if it's a passive diffusion, the movement of molecule from high concentration to low concentration using the cell membrane. If it's a facilitated diffusion, the movement of molecules from high concentration to low concentration using an integral protein. On the other hand, active transport is the transfer of molecule from one side to another side using ATP. Okay, let's move on to the next topic, which is the culture media, or in other words, what we're going to feed to the bacteria, the food for bacteria. Okay, culture media. So by definition, any nutrient prepared in the lab for the growth of microorganism is known as a culture media. So usually culture media contains the right amount of or right kind of nutrients, pH, moisture, and oxygen. So culture media, it can be liquid, or we call it a broth, or solid, we can call it a agar. So the broth media are usually prepared by dissolving nutrients in the water and then we autoclave to sterilize them. On the other hand, the solid media are made by adding solidifying agents such as agar to the liquid medium. Okay, what is agar? So agar is a galactose polymer obtained from cell walls of some species of red algae or seaweed, which is found in Eastern Asia and California. Now actually this agar is an edible agar that we use at home to make jelly. And this agar has a particular features that are useful for their bacterial work. So usually this agar provides a solid surface for the bacteria to form colony. And therefore it is very useful to prepare a pure culture and then to describe colonial characteristics as well as very useful for the bacterial enumeration. Okay, agar has like a few important properties that make them suitable for bacterial work. So number one, only a few microbes or microorganisms can degrade the agar. So which means when you're growing the bacteria or any microorganism, the agar stay as it is. And number two, the melting point of agar is almost like water, which gives a good flexibility for culture incubation. And number three, agar stays as liquid at temperature as low as 40, so which make them suitable for the addition of a drug or heat sensitive supplement into the culture media. So in general, there are 8 types of culture media, which is including defined media, complex media, selective media, differential media, enriched media, enrichment media, anaerobic media, and transport media. Okay, the first one is the defined media. So as the name tells us, so the exact chemical composition and their amounts are known in this media. So as an example, as shown here. So many organic growth factors are also added to enhance the growth of the bacteria. Okay, the second one is the complex media. So this media contains some ingredients of unknown composition and or concentration. For example, yeast extract and beef extract. So usually yeast extract and beef extract, they supply amino acids, vitamins, growth factors and etc. to the media. Okay, the third one is the selective media. So as the name tells us, this media favors a particular bacteria and then did not favors the rest. So usually it's used to growing a specific type of bacteria. For example, 7% NaCl medium for the Staphylococcus. So this media will enhance the growth or favors the Staphylococcus growth and then will suppress the growth of unwanted or other bacteria. Okay, number 4 is the differential media. So differential media are used to distinguish between different groups of bacteria on the basis of their biological characteristic. And the substances are often used to differentiate the bacteria, including the carbohydrate salts, dyes, and chemicals. Okay, this slide summarizes defined, selective, and differential media. So nutrient agar is a defined media. So it allows the growth of E. coli as well as Staphylococcus aureus. Meconic agar, it can function as a selective media. So when you strict E. coli and Staphylococcus aureus, is only allow the growth of E. coli and then it suppress the growth of Staphylococcus aureus. So therefore, Meconic can function as a selective media. And then on third here is a Meconic agar again, but then here it functions as a selective media because E. coli form like a rosy pinkish colony, but then Salmonella it form a amber colonies. So therefore you can use this media to differentiate between E. coli and Salmonella by their colony appearance. Okay, the fifth one is the enriched media. So enriched media are the media are supplemented by blood or other rich nutrients to promote the growth of fastidious microorganism. An example is the blood agar, where the media is supplemented with real human blood. So if it is chocolate agar, it has been supplemented with cocoa powder. Okay, 6 is the enrichment media. So this is the media which promotes the growth of a particular organism by providing essential nutrients. And this media rarely contains inhibitory substance to prevent the growth of normal flora competitors. For example is selenite F broth. So this media favors the growth of Salmonella but only slows down the growth of normal competitors like E. coli. Okay, the seventh one is the anaerobic media, or also known as reducing media. So this media contain reduced amount of oxygen by using chemical compounds such as sodium thiolate, and it is suitable for culturing anaerobic bacteria. An example of media is Robertson cooked meat, which actually made with meat, and also thiolate medium. Okay, the last one is this transport media. So transport media is a safe and appropriate way of carrying samples from short or long distances, and some are designed to speed up the growth of microorganisms in the sample and helps the isolation, and some are designed to slow down the growth of contaminants that are coming from normal flora. An example of transport media is a buffered glycerol saline. Okay, thank you very much for your attention. So please post your questions in the e-learn, and then I'll see you tomorrow at 11.